Welcome everybody. My name is Juami van Gils and today I would like to tell you something about frontotemporal dementia. Frontotemporal dementia is an early onset neurodegenerative disease which is caused by damage in the frontal and temporal lobes of the brain. It has a lot of comorbidity with other neurodegenerative diseases such as Parkinson, Alzheimer or ALS, for example. It's a very complex disease with many different subtypes. Clinically, it can be distinguished in a behavioral and a language component, whereas genetically it's often distinguished in a tau, a TDP and a non-genetic group. So the question we wanted to answer was which molecular mechanisms actually underlie this disease. To do that, we got 26 post-mortem samples from the medial frontal gyrus of the brain, which we then used histological staining to characterize whether they were tau, TDP, or a control group. At the same time, we extracted the proteins from these samples and performed mass spectrometry to determine the protein expression levels in these samples. Now, the first thing we see in this clustering figure, so at the top, you see the clustering of the different sample groups, and on the left you see the clustering of the different proteins. Now on the top you immediately see that the tau and the control groups form very well defined clusters, whereas the TDP group is more heterogeneous. And the same thing we see with the differential expression analysis. So you see the significance of the differential expression analysis of the tau versus control, TDP versus control, and tau versus TDP on the left. And so the more dark blue, the more significant differential expression analysis was. And also here you see the biggest difference between the tau and the control group and a weaker signal in the TDP. And so for the rest of this talk, most of the results I will show are for the tau versus the control group and not so much for the TDP. Now, to determine what is going on in the disease, we performed a co-expression analysis. So what we did is that we calculated the correlations between the protein expression levels of all the proteins. So this is regardless of any functional annotation or the sample groupings. Now using these correlations, we get different modules of co-expressed genes, after which we then perform co-term enrichment analysis on each of these modules to determine which molecular functions are enriched within each of the modules. Now note these modules are quite big, so every module is typically associated with multiple molecular functions. Also, because these modules are quite big, we wanted to know which proteins were at the core of each of the modules. And so what we did is we created a network of each module based on the protein correlations. We then added the log2 fold changes of the tau versus the control group, and then performed a hierarchical hotnet to identify the most dysregulated subnetwork within each of the modules. So what we see, we found 17 different modules, and I show you one here as an example. This module was enriched in proteins involved in vesicle-mediated transport. And what we see in the subnetwork here is that most of the proteins in the subnetwork have a lower expression in tau versus control, except for this one, which is called PTBP1, which has a much higher expression in tau versus the control group. So we looked a bit at the function of these different proteins, and a lot of them make sense from a brain disease point of view. So for example, PTBP1 regulates the splicing of tau, which affects its aggregation propensity. Now, tau aggregates are found in many different neurodegenerative diseases. So a protein that actually affects the aggregation of tau could be very important for the disease progression. So the next question is, how do we know that the modules that we found have real biological meaning and are not just a chance finding? So for that, we developed a new validation method where we compare our data set to a validation set. And what we do is that we calculate the fraction of proteins in the module that have a log2 fold change expression in the same direction. So either both up in tau versus control or both down in tau versus control, and have a p-value lower than a certain threshold. Now we calculate this fraction over a number of different p-value thresholds going from 0 to 1. And then we do the same thing in a permuted setting. So we shuffle the labels in one of the two data sets, calculate the same fraction, and then repeat this 1000 times. So the permuted setting gives this distribution that you see in gray, whereas the actual comparison gives you this black curve over here. And so what you hope is that if the black curve consistently stays above the distribution in gray, then you have a real biological signal in your module. If it doesn't, then it's probably a chance finding. And so we found a validation set of an independent cohort where they looked at the difference in protein expression of tau and control of patients, both in the frontal and the temporal region of the brain. And so you have two samples per patient. And so effectively, we actually have two validation sets. And so going back to our module enriched in vesicle-mediated transport, at the top you see the network that I showed you before. And at the bottom you see the subnetwork of the validation set. And what you can see is that these are very similar. So most of the proteins have a lower expression in tau versus control. And PTBP1 again really jumps out as the protein with a higher expression in tau versus control in this module. You can also see on the right that the fraction of proteins that is similar in both datasets is really higher in the actual comparison compared to the per mutant distributions. And so we could really validate this module. Now in total, I show you a couple more. We could validate 8 out of the modules in the frontal dataset and 10 of the modules in the temporal dataset. And so among the modules that were validated, we can find modules enriched in post-synapse organization, oxidative phosphorylation, vesicle-mediated transport, and phenylalanine tRNA ligase activity as an example. And these are all functions that are highly relevant for brain disease. And so we can really find biological signal using our method. So in conclusion, we see that FTLD tau has a more defined molecular profile than the TDP group. And we can find these different modules that are enriched in biological functions that seem relevant for brain disease. So we really have some meaningful findings here. We can also highlight the strongest dysregulated core in each of the modules that we find. And finally, we developed a method to validate the identified modules. And so with that, I would like to thank everybody involved in this project. And I would like to thank you for your attention. I mean, if you have any questions, I would love to see you at my poster.